Good morning, students. In the last lecture, we have discussed about the different techniques used in recombinant DNA technology. So today we will discuss about the crop improvement. So as we know, in the case of crop improvement, there are various technologies used. So firstly, we will discuss about the traditional method in crop improvement. So if we talk about the traditional method, it is plant breeding technology. It was traditional method in which we used to choose different varieties which are having specific characters, and the desired character we want to introduce, we used to make cross. So after cross, many crosses, the successful variety we can identify through the RAPD or so many techniques, SSR markers, by using that techniques. But in the case of plant breeding, there is something that used to take longer period. But in the today's time, in the current scenario, the technology used for crop improvement is called recombinant DNA technology. It is also called RDT, and it is the current approach for improving any crop. So as in this lecture, I am introducing you about the crop improvement, why there is a need of crop improvement. So in the crop improvement, as we know, if we take about the example of golden rice, for example, in current scenario we are eating brown rice or golden rice. The concept behind it, because as we know, rice is having more starch, more carbohydrates, and that is not good for our health. So for the better health, for the better characters, nutrition, we can choose golden rice or we can choose brown rice. So in that rice variety of crop, there is more protein is there. How that can occur? That is occur through recombinant DNA technology, that is approach of biotechnology. In this we can introduce a gene, specific gene in the RDT. What we can use? We can use a specific gene which codes for desired protein. So for example, I'm talking about the golden rice or brown rice concept. So we will take which specific gene that codes for desired protein which occurs in brown rice or golden rice. In that gene we will introduce through an RDT process in particular variety of which rice. So when we will introduce this gene into particular variety of rice, then the variety will be more proteinaceous, and that is called brown rice or golden rice. After that, I want to discuss more. If we talk about crop improvement, we are talking about RDT. In RDT, we are using a vector. What we need? A vector that can be a different plasmid vector, that can be a plasmid vector, that can be a yeast vector. It depends upon the size of DNA we want to introduce in the host cell. So in the vector is there is a site for insertion of particular DNA, or we can say specific gene which will express into desired protein. So what we are taking firstly in the RDT technology, we are choosing a crop where we want improvement. So if I am talking about any plant, any crop, means any plant or crop where we need improvement. For example, I have talked about rice, golden rice concept. Now I'm taking another example. As in the Himachal Pradesh, we know apple is, large production of apple is there, more production of apple is there. So we need different varieties of apple who are more delicious, whose production is yield is more than other varieties. So for that, on the basis of that, or if we are taking another character, like we want to produce apple variety in the lower Himalayas. In the upper Himalayas, in the high altitude, there are so many varieties are there, but in the lower Himalayas, if we want to introduce that varieties of apple, we need specific characters so that that varieties can be grown in the lower Himalayas. So for the growth of that variety, or for more fruit of that variety, what we are taking? We are taking a specific gene which shows that character. So how we can introduce that character into apple, variety of apple? We will take that DNA of variety, best variety, and then we will take that DNA or gene of that variety, best variety, who are having best characters, best characters in the field of like apple fruit, size, more production, more yield. These can be the different characters. So after taking that gene, we will introduce this gene into a vector, and after this vector, we will introduce into the host plant, where we need that character. Okay, I think you understood about the host plant means apple. Here we are talking about apple. So now after introducing this vector is having specific gene. This is the area having specific gene, that is the specific DNA. Now this vector will be introduced into host plant, and after the host plant can be grown into the field. Now this variety can be grown into the field or into the specific area. Second point is that, firstly what we have done, we have chosen a variety that we want to grow in the lower Himalayas, the variety of apple. Now we have put the specific characters, the best characters, what we want to see in that variety. Then we have used recombinant DNA technology, in which vectors play important role. Through vector we have introduced a particular specific gene into that variety of apple. Now that variety is grown, and the next step is multiplication. When we got the variety with the specific characters, the next step is multiplication. So when the multiplication we want to do in the apple of variety or any of variety, like rice, apple, anything, desired variety of plant. So what we will do? The next technique is called tissue culture. It is also called plant tissue culture. Okay, so we will discuss about the next technique, plant tissue culture, that is useful in the case of crop improvement. So for the crop improvement, we have talked about RDT, we have talked about the plant breeding, both can collectively we can do what technique is called plant tissue culture. Basically for the multiplication. So firstly I want to discuss, if we want to improve any variety of crop, we can do it by two approaches. As we discussed, in the traditional method we can use plant breeding, and in the current approach, that is called recombinant DNA technology. Why it is called recombinant DNA? Because if we are taking any vector is having own DNA that can self-replicate. When we are putting a specific gene, or we are using the specific gene and we are inserting that gene into the vector, that vector can be further transformed into the host cell where we want desired characters. So after using this RDT technique, we can further go for tissue culture. Why there is a need of tissue culture? The second question is that as we have grown with a new variety that is having a specific characters, that is having everything what we desire. Now we want that variety to be distributed among the farmers so that they can be economically strong. So after producing the best variety having the specific characters, we will move for plant tissue culture technique. So plant tissue culture technique is useful for farmers. How it is useful? Because in this technique we can multiply thousands of plants in the couple of months. So in this technique, it's self-explaining that plant tissue culture. In this technique, what happened? In this technique, we can use the different parts of plant, that can be cells of plants, that can be tissues of plants, and these tissues or cells we use to grow that tissues in the nutrient media under the sterile conditions. So basically, what is plant tissue culture? It is a technique in which we use different tissues or cells of plants and that can be grown into the nutrient media under the sterile conditions. So in this plant tissue culture technique, how it is useful? Because in this technique we can multiply the thousands of plants in couple of months. So in plant tissue culture, exactly what we need? We need a nutrient media, we need plant growth regulators, what we need plant growth regulators, we need sterile conditions, if we talk about sterile conditions, we need different instruments that can be called autoclave, LAF, it is called Laminar Airflow, and like we need hot air oven, etc. that is useful for sterilization process. So if we talk about tissue culture technique, there is another specific need of plant cells, or we can say tissues. What we can take? Plant cells or tissue that can be grown in nutrient medium under sterile conditions. So plant cells or tissue can be grown on nutrient medium under sterile condition, that is called plant tissue culture. So why there is a need of plant tissue culture? As I have discussed earlier, for the crop improvement, if we are generating a new variety that is having the best characters, if we want to multiply that variety, if we want to take more yield, what we will do? We will do plant tissue culture of specific plant of the desired plant. So if we are talking about, first of all, I want to let you know about the tissue culture technique, plant tissue culture technique. What we will take? We will take tissues or cells, upper part part, that shoot with a leaf, we will take a medium, nutrient medium, that is called MS media. What is MS media? It is also called Murashik scoop medium. That is on the basis of, the name is happen on the basis of the different scientists who work for this media, and it is called Murashik and scoop medium. So in the MS media, we will take that tissues and put into this media, and where this will occur, this all will occur under sterile condition, that is laminar airflow. If we talk about laminar airflow is an instrument in which there is a use of HEPA filters, high efficiency particulate air filters. If we talk about HEPA filters, this used to, it is like a cake which used to entrap the different microorganisms who used to do contamination. So in the MS media, we want to grow only these tissues. We want to grow only these tissues, these cells, so that they can be grown under this MS media. After the full growth, where it will grow? It will grow under specific conditions. Specific conditions in growth room. In the growth room. What is growth room? If we talk about the growth room, first of all, what we need? A need of desired plant, which we want to multiply. After that, we will make a MS media. What is MS media? It is the media which are having all essential micronutrients, macronutrients, which are useful for the growth and development of that plant. So after preparing MS media, it will be autoclaved. MS media will be autoclaved. What is autoclave? If we talk about autoclaves, it is also instrument. It is an instrument used for sterilization process. In the sterilization process, if we want to use MS media for the growth of plant, it should be microbes free. It should be contamination free. And how it can be possible? It can be possible by using autoclave. So in the case of autoclave, the basic principle behind autoclave is high steam under pressure. In the autoclave, the temperature is about, it is an instrument in which temperature is about, we used to kept 121 degrees Celsius, and 15 psi is about the pressure. So that all the microbes which are inside the MS media, during preparation of MS media, if is any there, so after using autoclave, there will be no contamination further. So we have prepared MS media, which are having all the micro and macro nutrients, which are useful for the growth and development of that plant. So first of all, we have used tissues and cells of plants. We have inserted these tissue and cells to the nutrient media, which is called MS media. And if you talk about MS media, it is Murashik and scoop medium, which is having all the essential micro and macronutrients. We also use, after that, the plant which is inside a flask, which is containing MS media, that will be kept in the growth room. It will be kept in the growth room. And what is growth room? Growth room is a room in which we will kept all the conditions, specific condition up to the mark what plant will require. We will maintain the day cycle, night cycle. We will maintain the sunlight, which is required. So by replacing sunlight, how we can replace it? By using tube lights. We will manage the humidity. We will manage everything, every specific condition we will manage in the growth room, so that the shoots and roots of the specific variety of plant can be developed. So it is in the plant tissue culture, we don't require soil. What we require? Artificial media. What we require? We require growth medium. We require growth room, where we can artificially generate or multiply the specific variety of plant. So again, I am going for the specific technique or the process. We are using tissues and cells into the MS media. We have inserted it. Where we have inserted? Inside a chamber. Inside a chamber, or we can say an instrument that is called Laminar Airflow. I have already discussed about it. It contains HEPA filter, and which used to stop different microbes from the air. So after using this Laminar Airflow, we will put these cells or tissues of specific plant into the MS media. Then that flask containing MS media and particular desired plant, we will shift into the growth room. And I have already discussed about the growth room, where all the conditions will be kept optimum. Means like tube light, the daylight, about the night cycle, about the humidity, about the air, temperature, everything will be maintained. A pH will be maintained through MS media. So everything what plant requires in the natural way, we will keep these conditions in artificial way in a particularly lab. Okay. So after using this growth room, after 30 days, after the 30 days, you can see the plant inside the flask. The MS media, it will be used all the nutrients for the growth and development, and you can see here the plant will be fully developed. Now there are two types of media. If we talk about MS media, there are two types of MS media. First is called shoot culture and second one is called root culture. So first of all, what we require? We want to proliferate that plant, the desired plant. So what we will use? We will use shoot culture. But we will use shoot culture so that the upper part of that plant can be fully proliferated. So after the fully proliferation of that upper plant, what we will do? We will take that plant from, in the laminar airflow, we will take this plant and put this plant again into what? Into the root culture media. Into the root culture media. As I have discussed you about the plant growth regulators. Now there is a role of plant growth regulators. What is the role of plant growth regulators? It is having specific importance for the growth and development of shoots and particularly roots. So in the shoot culture, we will require different plant growth regulators. What are plant growth regulators? They are auxins, cytokinins, gibberellins. So these plant growth regulators are used for the growth of various shoots and roots of particularly desired plants. So as we have discussed in this process, you can see we have taken tissues, cells of particularly desired plant. This is a desired plant which we want to distribute to farmers. Desired plant which are having specific characters. As we have discussed in the last slide, that we have prepared a new variety of plant which are having specific character, like we are talking about the brown rice. The rice variety is having particularly gene which is more rich in proteins. So basically the function of plant tissue culture technique is to multiply the different plants, thousands of plants in couple of months. So in this plant tissue culture technique, as you know that we are taking the desired plant which are having specific characters, we will take the tissues and cells of that specific plant, we will grow these tissues in the MS media. As you know what is MS media? MS media basically is a nutrient medium which is having specific micro and macro nutrients which are useful for the growth and development of the plant. So by using these cells or plants in the MS media, this process will be occur in the laminar airflow. As I have discussed earlier, that laminar airflow is the instrument which is used for the sterilization process. It provides a workstation, a specific contamination-free workstation where there is no microbe from the outside. How it is possible? It is possible through the use of HEPA filters in the laminar airflow. What are HEPA filters? These are the high efficiency air particulate filters which used to entrap microbes from the outside environment. So by using this laminar airflow, it will provide a workstation for the transferring of these tissues to the flask which are having MS media. And what is MS media? It is basically a nutrient media. As I discussed earlier, it contains particularly micro and macronutrients which are useful for the growth and development of the plant. In the MS media, there is also, we call it agar. It is used. It provides a saltification. It makes the media a saltified media like gel, and agar is used for this function. Also, I have discussed that there is a use of plant growth regulators. As I have discussed earlier, that what plant tissue culture requires? It requires a nutrient medium that is called MS media. What is MS? MS is Murashik and Scoop medium. These are two of the scientists. We have prepared this media for the growth and development of plant inside artificial room, or we can say in the replacement of soil. So by using this nutrient medium, we can grow the specific variety of plant. We can use plant growth regulators. What is the function of plant growth regulators? These regulate the growth of plant. That can be a shoot or that can be a root part. So this all process will be occurred in the growth room. When we will put the plant part in the MS media, that flask we will grow, that flask we will put in the growth room, and the plant will be grow in the under the specific conditions, that is pH, that is temperature, that is the humidity. All will be provided, like light, all will be provided in the growth room, artificial conditions under the optimum conditions. So the specific instruments used for the sterilization process in the plant tissue culture technique are autoclave, hot air oven, and as I have discussed, laminar airflow. So in this technique, as I have discussed, that we will put the tissues and cells of plant inside the media, MS media. There are two types of MS media. If you want to proliferate the upper part of the plant, that is shoot part of the plant, we will use shoot culture, which is a full concentrated MS media having full stalk cultures, having full micro and macronutrients concentrations. But in the case of root culture, when we want to grow roots of that plant, we will take root media, and in the root media, the concentration of the MS media is kept half. Why it is kept half? Because the plant comes in the stress, and because of that stress, it generates its roots for further development. So, after shifting or transferring the cells into MS media, this flask will be moved to a growth room, as I have discussed, growth room, where the conditions will be kept optimum, like pH, temperature, humidity. After the growth room, that plant will be, when fully grown by the shoots, that part will be now transferred into the another flask who is having root culture media. So in the root culture media, as you can see here, the flask is containing root culture MS media, and where the concentration of this media will be half. Why it is half? Because the plant will become in stress and it will start generating roots in the plant, or after that putting or shifting the this plant in the root culture media. After the 15 days or 20 days, when there is a full consumption of that media will be there by the plant, then that plant will be shifted to small pots. It will be plant will be shifted to different environments so that it can be known with the outer environment. So it will be kept like a small baby, so that gradually it starts to known with the conditions of outer environment. So after putting that plant into the pots, small pots, it will be now shifted to shading nets, where it will gradually known with some conditions of outer environment. Then this process is called primary hardening. This process is called primary hardening. From a flask to the pots, then pots will be shifted to a shading nets. Then shading nets, this process is called primary hardening. That hardening, we are moving the plant to some but little hardened conditions. Then the plant will be again moved for outer fields, and that is called a secondary hardening process. And after that you can distribute that plant among the farmers. So this is overall technique that is called plant tissue culture. So in this technique, what we have studied? We have studied the role of growth regulators there, that is auxins, cytokinin, gibberellins. We have discussed about all the things that is useful for the growth and development of the plant. So in today's lecture, we have discussed about the overall technique, crop improvement. Why there is a need of crop improvement? Because if we want to introduce new variety and we want to multiply that variety, then we need plant tissue culture technique, we need recombinant DNA technique. So in this lecture, we have concluded about all these things. Thank you so much.